In this last video of the cell cycle lectures, I will be discussing about the stages of meiosis. Now, meiosis is a special type of cell division in eukaryotic cells in that the resulting daughter cells are actually all unique from each other. So this is in contrast to mitosis, whose resulting daughter cells, which are only two, are identical to each other. So let's start first with what happens to a mother or a parent cell in the prophase stage of meiosis. So we can see here on the first illustration that there is a certain stage or portion of prophase 1 called leptonema, wherein the chromosomes actually start to condense. And you can see the formation of bead-like structures along the length of the chromosomes. So these are the chromatin structures from interphase now condensing slowly into their discrete structures, and we now see these structures in leptonema. And then later on, the chromosomes actually do something that didn't happen in mitosis. They pair with their homologous pair or their homologs. So, meaning to say, a chromosome of similar position of centromere from the father and similar locus or loci of genes from the father will pair with a chromosome of similar position of centromere and loci of genes but from the mother. So that's what we see here between the blue and red chromosomes pairing up, forming a bivalent. So this is now the zygonema stage of interphase. And then finally, the replication of the DNA becomes more obvious now, as we notice now in the pachinema stage that each chromosome is actually made up of two sister chromatids sticking to each other. So the homologous pair will now appear to be four parallel or sort of parallel clumps, and we call this now a tetrad. So this is now the pachinema stage. And then the tetrads will then sort of cross one or more of their ends, as we see here in diplonema, forming a chiasma. So this is now the chromosomes preparing to undergo a process called synapsis. First with the formation of chiasma or chiasmata in plural. And then as spindle fibers form, they actually move the chromosomes which are still attached as tetrads about, and we call this the diakinesis stage of prophase. Now take note, this is prophase 1 because there's actually a part 2 which also has a prophase. Now after diakinesis and homologous pairs have stayed with their pair, with their homologs, as we see here, they will then slowly travel towards the equatorial plane or the metaphase plate of the parent cell. So this is prometaphase 1. Then in metaphase 1, we notice here that the homologous pairs stay paired with each other and their chiasma stay as is. So this is because of the process of synapsis that's going to be happening still. And in anaphase 1, the spindle fibers that have attached to the kinetochores of each of these chromosomes will pull one homolog away from the other. So a homolog will go to one pole while the other pair of the homolog will go to the other pole. But because there was a chiasma and crossing over happening, okay, as we see here in metaphase 1, the chromosomes or the homologs actually sort of swap portions of themselves. As we see now, we have a chimeric homolog. We have a homolog that's a majority blue, and then there's a small portion that's red, and a homolog that's a majority red with small portions of blue. And that's a result of synapsis. And so the homologs now travel to opposite poles and become contained in two daughter cells, as we see here in telophase 1. Okay? And notice that in telophase 1, as it proceeds to prophase 2 now, okay, the two daughter cells are not identical anymore. Just look at the composition of their chromosomes. They both contain one homolog each, but the homolog for both of them are different in terms of the colors. So prophase 1 will happen right away after telophase 1. There's almost no interphase. Okay. Sorry, prophase 2 will happen right away after telophase 1. And we can see here on the top portion what happens to the first daughter cell for prophase 2. Okay. The chromosomes will again migrate to the center if they haven't yet, and then align at the metaphase plate. So the chromosomes now, which are just sister chromatids attached to each other, now have aligned at the metaphase plate. The same is true for the second cell here at the bottom. And then the spindle fibers attached to the kinetochores again pull, this time, the sister chromatids apart from each other. Okay? And we sort of arrive at 4 daughter cells in total here at the end after telophase 2. And all of these cells are different from each other. Again, just look at the colors of their chromosomes. And notice also each of them just contain 2 chromosomes each from their parent, which had 4 chromosomes. So they now have half of the amount of chromosomes or number of chromosomes. And they are haploid as opposed to their parent, which was diploid. And really, the purpose of meiosis is to produce what we call as gametes or sex cells. And gametes can occur as sperm or egg in animals or spores in fungi and plants. So this is an overview of what actually happens in the formation of gametes. We call as gametogenesis. On the left here, we have what we call a spermatogenesis, the formation of new sperm cells. And on the right, we have oogenesis. On the spermatogenesis, we can see here that the sperm cells, which are haploid, actually come from a diploid cell called a spermatogonium. The spermatogonium matures into a diploid primary spermatocyte. And the primary spermatocyte undergoes meiosis. And after meiosis 1, the 2 daughter cells are known as secondary spermatocytes, and they are already haploid. Okay? So notice just after meiosis 1, the resulting daughter cells are already haploid. And then these 2 secondary spermatocytes furthermore divide in meiosis 2 to become 4 haploid spermatids. And the spermatids mature in a process called spermiogenesis to become mature sperm cells or spermatozoa. For the egg or ovum formation or oogenesis, the original diploid cell is the oogonium, which grows and matures to become a diploid primary oocyte. And then in meiosis 1, the primary oocyte becomes a secondary oocyte, and because the cytokinesis is not equal in terms of distribution of cytoplasm, one of the daughter cell, which is the first polar body, is small, a lot smaller than the secondary oocyte, and is actually non-functional as a gamete. So it stays as is. So the secondary spermatocyte then will undergo another stage of meiosis, meiosis 2, to form the ootid and a second polar body, because again, the division of the cytoplasm is not equal. And this ootid will mature to form the egg cell or the ovum. So all of these happen in the gonads or the sex organs. In the male, that's the testis. So we can see here another diagram of spermatogenesis. Then here's how it happens in the ovaries for females. So that's the sex organ for the females where oogenesis happens. And notice here that even as an embryo, females will already have primary oocytes in their ovaries. And they are already accounted for. Okay, they are already stored there. And at puberty, this primary oocyte will start undergoing meiosis. So meiosis 1 is finished, producing the first polar body and the secondary oocyte. And the secondary oocyte is somehow in arrest of meiosis 2, meaning to say it stops there instead of continuing to meiosis 2. The trigger to continue to meiosis 2 is the contact of a sperm cell to that secondary oocyte. Now, when a sperm cell fertilizes or is in contact now with that secondary oocyte, the secondary oocyte continues to meiosis 2 and becomes now the fertilized egg when the nucleus of the sperm, which has attached to it, unites with its nucleus. So here's a comparison of mitosis and meiosis. Mitosis actually preserves the number of chromosomes. Okay? So if the parent cell has 4 chromosomes or 2 pairs of chromosomes, the daughter cells, which are only 2, should also have 2 pairs or 4 chromosomes each. Meiosis is reductional in nature, meaning to say it halves the number of chromosomes and genetic material. So that if the parent cell has 4 chromosomes, then the 4 new daughter cells in the case of sperm, or 2 or 1 daughter cell in the case of the egg cell, will only have half or 2 chromosomes.